Okay, the next cells we are going to talk about are going to be the leukocytes or white blood cells. We already talked about it in the sense that we said that the number of white blood cells that we have in our body are going to be approximately from 5,000 to 10,000 per microliter. The job of the white blood cells is basically to protect our body. Protection of body consists of multiple different steps that must be taken by all these different kinds of cells that we have inside our body that are going to help us with protecting our body against foreign substances. To make things easier, there are bacteria, viruses, parasites, allergens like pollen, for example, and they all are different. So therefore, that also may explain the different kinds of white blood cells that we are supposed to have, because we have so many variations of substances that are outside our body that when they come in contact with us will produce reactions and diseases in our body. Therefore, our body has to have different types of cells in order to fight these different kinds of triggers that may produce disease in our bodies. So we said the white blood cells are going to be divided in basically two main groups. We said the granulocytes and the agranulocytes. One characteristic that red blood cells have is that they do not have nucleus and they don't have organelles. On the contrary, white blood cells, they do have nucleus as well as cell organelles, as you can see in this figure. Okay, so on the types of leukocytes, we said that we have the two main groups, granulocytes and agranulocytes. The granulocytes are going to be neutrophils, basophils, and eosinophils, and the agranulocytes are going to be obviously the monocytes and the lymphocytes. The names vary obviously depending on the type of cells that you have and the amount of presence of granules or not, but the names most of the time comes from the type of stain that is being used in order to differentiate them when we try to put some color in these cells. And that's where the names come from, such as basophil, eosinophil. Okay, so let's take a look at these type of cells with a little bit more detail. For example, we have right here the granulocytes. As we said, granulocytes are going to be neutrophils. Right here, neutrophils, eosinophils right here. And the next one is going to be obviously neutrophil. If we scroll down, we're going to see the next one is going to be basophil right here. So I guess I already show you the other one. Yeah, neutrophils, eosinophils, and then the last one is going to be basophils. So these are the granulocytes. Since they are granulocytes, obviously they're going to have granules inside with multiple different functions. Then we have the agranulocytes. We said the agranulocytes are going to be the lymphocytes right here, lymphocytes, and the monocytes. If we keep scrolling down, you're going to see that the next one obviously is going to be the monocytes. So instead of reading all the text that we have in here, might as well just take a look at the table where you have a summary of all the information that you have right there. So if we look at the table, the first one that we have in here is going to be neutrophil. As you can see in here, every single time you see a blood smear, which is the blood that you get, a couple of drops of blood that you get, you put it in a slide, a microscope slide, and then you cover the slide with a cover slip, and you put it in the microscope, then there's a chance or there's a probability that the cell that you're going to be seeing in there is going to be a white blood cell. What white blood cell, 60 to 70% of the time, is going to be a neutrophil. That's what this means. So then, the most abundant one, neutrophil is going to be, the most abundant white blood cell that you may find in the blood is going to be neutrophil. What are the functions of the neutrophil? It phagocytizes bacteria. Obviously, phagocytizes means eats the bacteria. And it's going to release antimicrobial chemicals in order to destroy them as well. These chemicals are the ones that you're going to find right here inside the granules. So a couple of characteristics that you're going to have regarding the neutrophils is that they are the most abundant white blood cell that you're going to find in the blood smear, as well as the characteristics or functions that they have, which is eats bacteria, phagocytizes bacteria, and releases antimicrobial chemicals. These chemicals are the ones that you're going to find in the granules. So those are some of the characteristics that you're going to find for neutrophils. The next one that you're going to have is eosinophils. It's not the most abundant, it's not the least abundant either. And we're going to see in a little bit. You find them more or less 2 to 4%. Let's take a look at the functions. The eosinophil, the main job is to phagocytize antigen-antibody complexes. Phagocytize again means what? Eating. In addition to that, also the release of inflammatory chemicals. You release these chemicals that you have in here and they're going to stimulate inflammation. And also they are going to phagocytize antigen antibody. An antigen is any substances that comes from outside your body inside, therefore producing a reaction in your body in order to get destroyed. The chemicals that you're going to have inside these granules, when they are released, they're going to weaken or destroy, for example, parasites such as worms. So when you have a parasitic infection, chances are that you're going to have an increasing number of eosinophils that you're going to find in your blood smear. So let's take a look at the other one. The next white blood cell is going to be basophils. You can hear the basophils, you're going to find them less than 0.5%. Then obviously that indicates that basophil is the least abundant white blood cell that you're going to find in a blood smear. What's the characteristic? As you can see here, it has a whole bunch of granules in there, right? Therefore, these chemicals that you're going to find in these granules are going to be mainly histamine, which is a vasodilator, meaning it stimulates the arrival of more blood, therefore more white blood cells can arrive in the area where you may have an inflammation, as it says in here, which increases the blood flow to a tissue. Also secretes heparin, which is an anticoagulant, okay, which promotes obviously the mobility of white blood cells. The next one, and now we go into agranulocytes. The very first one of the agranulocytes that you're going to have in here is going to be lymphocyte. The number of lymphocytes is 25 to 33% of white blood cells that you may find in the blood smear. The main job of a lymphocyte is actually very abundant. It has multiple different functions. One of the most important ones that you need to remember is that it produces, let me see if they put it right here in this summary. There you go. It produces antibodies. Very important, as we're going to be talking antibodies in many different chapters during the course. So you have to remember that the lymphocytes are the ones that are in charge of producing antibodies. Later on, you will see that there are two types of antibodies. There are two different types of lymphocytes. One lymphocyte is going to be lymphocyte B, and the other lymphocyte is going to be lymphocyte T. The lymphocyte B is the one in charge of producing the antibodies. The lymphocytes T are, for example, the ones that are in charge of destroying cancer cells or cells infected with viruses. So, two very important concepts that you need to know is that for the lymphocytes, you're going to have lymphocytes B and lymphocytes T. One of the main characteristics of the lymphocytes B is that they produce antibodies. One characteristic of the lymphocyte T is that they are going to destroy cancer cells, cells infected with viruses, as well as other cells that may come from the outside of the body. The next one is going to be a monocyte. Monocytes are approximately 3 to 8% of the white blood cells that you find in a blood smear. One very important characteristic is that the monocyte, once it leaves the blood, it will turn into a macrophage. The macrophage is a cell that is going to actually phagocytize foreign substances as well as foreign foreign antigens that you may find in your tissues. So as you can see here, it says it differentiates into macrophages and phagocytizes pathogens that comes from obviously outside your body. And that's the last cell that we have. Where do white blood cells come from? The leukocyte life history. Well, like any other cell in your blood, they come from the bone marrow. Same way like in the red blood cells, the cell that is going to produce the white blood cells is going to be a stem cell. The stem cell is going to then differentiate into other type of cells, and eventually these other type of cells, through a series of changes and variations, eventually are going to turn into the basophils, eosinophils, neutrophils, monocytes, etc., etc. Right here you have B lymphocytes and T lymphocytes. As I was telling you, the lymphocytes, you have two main kinds, which are going to be B and T lymphocytes. You don't need to know all these steps in here. As a matter of fact, I mean, you can just disregard the steps. You need to know that the white blood cells are created in your bone marrow by a stem cell. This stem cell differentiates into multiple different lines of cells, and each line of cell eventually will provide you with the different kinds of white blood cells that we have in our body. If you keep going down, you're going to see different conditions in which you may have an increase or decrease of white blood cells. If you increase the white blood cells, then you have a disorder called, for example, leukemia. If you have low number of white blood cells, then you have leukopenia. In what cases you can have leukemia? Obviously, leukemia is cancer, right? Way too many cells. And in the case of leukopenia, is when you don't have enough white blood cells, such as, for example, in the case of AIDS. In the case of AIDS, the white blood cells are going to get destroyed. And then because of that, obviously, the number of white blood cells are going to be reduced in this kind of disease. One thing that you need to differentiate is that when you have an infection, the number of white blood cells will increase, obviously as a response to the presence of foreign cells inside your body. But this increase is an increase of normal cells in shape and form, and the increase is not that high. Like, for example, we said the number of white blood cells is from 5 to 10,000. So when you have an infection, these cells are going to jump maybe to 10,000 white blood, to 20,000 white blood cells. That's a normal response because of the presence of an infection. It's at 20,000 cells that they have normal shape, normal form, and they have the same characteristics like any other cell. When this number is not 20,000, and for example the number of cells is 100,000, that's when you have leukemia. And in this case, when you look at each one of these cells, you're going to see that the characteristics, such as shape and form, the size of the nucleus, is completely different than the normal cells. So remember, in the case of an infection, you were going to have an increase of white blood cells. But the increase is not going to be that much, maybe 20,000, 25,000. In the case of cancer, the number of cells also increases, but it's way higher, like 100,000. And the characteristics of the cancer cells are not going to be the same as the characteristics like you have in a normal cell. Right here, you have some white blood cells in here. Right here is one, another one, another one, another one. If you compare these cells, you have only 4. Here, you have a blood smear of a person that has cancer. In this case, leukemia, for example. And here you can see that the cells are way too many. Not only that, if you look at the sizes, look at this one, and look at this one. Size is different. Look at the shape. See the shape of this one? The shape of this one right here? So that's the characteristic of cancer cells. In this case, leukemia. Different shape, different sizes, the size of the nucleus is different, etc., etc. So I hope that this is clear. This is like 100,000 in the case of leukemia with different cell shapes and things like that. In the case of an infection, this number of cells is going to increase, but you see, for example, here you see like 4. Maybe you're going to find 6. And that indicates that you have an infection, but the shape and the characteristics of these cells are going to remain the same, which is a big difference with this one that you have right here.